So it's my pleasure to introduce this next session, which is genetics and future therapeutics. And we have experts in the fields, Dr. Wiggs, Liu, and Ross. And the learning objectives for this are to recognize the strength of GWAS studies to identify glaucoma risk alleles, recognize how glaucoma GWAS data is being used to understand glaucoma pathogenesis, and then recognize the challenges and opportunities for gene therapy or therapies to treat glaucoma. So with that, I'd like to invite our next speaker up, which is Janey Wiggs at Harvard Medical School and Mass Eye and Ear.
Thanks, Abe, and thanks to the organizers for inviting me back after, I think, 8 years was the last time I spoke in the Fast Track in 2017. So I decided to focus my update on that interval rather than going all the way back to the beginning. I've also heard from others that the speed on this timer is warp speed and that it goes by a lot faster than you think. So I'll be doing my best here. I have some disclosures, but none of those are related to this short presentation. So just to start with the background in terms of glaucoma genetics, it probably comes as no surprise to any of you that glaucoma is highly heritable. In fact, is one of the most heritable of all human disorders. But when we talk about glaucoma inheritance, it's important to keep in mind that there are a couple of different flavors or different types of inheritance. So we have the early onset glaucomas, like the childhood glaucomas, primary open-angle glaucoma, juvenile open-angle glaucoma, anterior segment dysgenesis syndromes. These are inherited as Mendelian or autosomal dominant or autosomal recessive traits. And what's important to understand is that these are mostly caused by single genes, single mutations in single genes, and those mutations in those genes have very large effects. So you have one of these mutations, you have the disease. There's no wishy-washiness about it. There can be some reduced penetrance and so forth, but we won't get into that. And so historically, these were actually the first genes to be found for glaucoma because there were techniques available to find them using familial linkage studies and large pedigrees. But more recently, with the advance of next-generation sequencing, whole exome sequencing, whole genome sequencing, now we can use smaller, sort of more typical pedigrees, actually, to analyze these families and to find additional genes. In contrast are the adult-onset glaucomas, and these are the ones that we actually talk about most of the time. I think today, so far, we did mention optineurin once in Dr. Meyer's presentation, but otherwise, mostly we've talked about adult-onset primary open-angle glaucoma, which is a disease with complex inheritance, as are the other adult-onset diseases. And so, in contrast to the early-onset diseases, these are associated with common DNA variants. These DNA variants influence risk, but individually, they have small effects. And you have just one of these, probably you're never even going to know anything about it. But in aggregate, if you have enough of these risk alleles, you can reach a disease threshold. These types of variants that we study are found through genome-wide association studies. Now, you might want to know, why do we even care about genes for glaucoma? And there are two kind of really important reasons. The first is that if you can identify these genes and these genetic risk variants, they can be used for genetic testing. And that's really important because you can identify people at high genetic risk, therefore high disease risk, ideally before they develop the devastating irreversible damage to the optic nerve that ultimately can happen in this disease. So you find a gene, you know it causes disease, you can test for it, identify people who carry mutations in that gene, and then know ahead of time that they're destined to develop glaucoma. That's very important, and I'm going to talk a little bit more about that today. The second is equally important. Characterizing these genes that cause or contribute to glaucoma can define disease mechanisms. Like we can know that a collagen is and there actually aren't any collagens, but we can know that, well, maybe one, but we can know that there's, a particular protein that is involved in the disease mechanism, and that gives us some clues about the disease mechanism, and also gives us ideas about what could be then targets of therapy. So that's kind of the holy grail in human genetics, and certainly also in glaucoma genetics. So I'm going to take you on a brief tour of genes for early onset glaucoma. I'm going to talk about a little bit about GWAS. I'm going to really focus mostly on genetic testing, and then a few odds and ends towards the end, and some ideas for the future. So first of all, these are the early onset glaucoma genes. So these were the first genes to be found, and it's kind of a short list, especially if you're familiar with the large number of genes associated with inherited retinal degenerations. But genes have been identified for all of these childhood types of glaucoma: primary congenital glaucoma, anterior segment dysgenesis, juvenile open-angle glaucoma, and familial normal tension glaucoma, which is what optineurin is a cause of. I think one thing we've learned as we've identified these genes is that a number of them are involved in developmental processes, so abnormal development of the eye, abnormal development of Schlemm's canal, abnormal development of the anterior segment pathway, fluid can't get out, you have high pressure, you have glaucoma. But what's emerging, I think, is an equally interesting a topic is the involvement of a number of these genes in extracellular matrix. So LTBP2, you heard about that today in the cats. Mycilin, which was the first gene to be found, EFEMP1, which we identified just a couple years ago, and thrombospondin. These are all genes that can have mutations and cause childhood glaucoma and the function in the extracellular matrix. So sort of knowing what an incredibly rich space the extracellular matrix is, with many, many proteins and protein interactions to think about, I think this is really ripe for future gene discovery. So adult onset diseases, this is where we do the genome-wide association study. This is the so-called Manhattan plot from one of the largest GWASs to have been done in the last couple of years. In the Manhattan plot, the genetic variants that are discovered through the GWAS are arrayed according to the x-axis with the negative value of the p-value on the y-axis. You probably all know this. Because you test about 1 million SNPs in a GWAS, you have to pass this really high bone Ferrari correction p-value, corrected p-value 5 times 10 to the minus 8, in order to show statistical significance. But, the field has been incredibly successful in identifying many, many, many genetic loci that are associated with primary open-angle glaucoma. Other types of adult onset glaucoma have also been studied using GWAS, and so this is just a list of kind of the last GWAS for the kind of common types of glaucomas. So the one I just showed you is for POAG, 127 loci. Recently, there's been a kind of a combined approach, POAG plus population samples of vertical cup to disc ratio and intraocular pressure, the so-called MTAG approach. That boosted us up a bit more to 312 loci for also kind of POAG-related loci. And then the largest angle closure glaucoma GWAS has 8 loci, not as many, and the exfoliation glaucoma GWAS has 7. Now, part of the reason why we have such a difference in number in terms of loci for these different GWASs is partly due to sample size. I think if you look at this table, you can see that, the POAG ones have really got dramatically huge samples, but it's not entirely that, because the angle closure and the exfoliation GWAS is also, are quite well sampled. So there's more to it, and one thing that there is more to it is the underlying genetic architecture of the disease. So this is the Manhattan plot for exfoliation syndrome, and the arrow there actually points to LOXO1, which the p-value there is actually minus 100. It's off the scale that the rest of the variants are on. And so 90% of the genetic risk in this disease is due to LOXO1. And you can increase your sample size 100-fold, and you're not going to find another variant probably that has an equivalent effect size as LOXO1. So it's partly sample size, but also partly the genetic architecture of the disease. So what can we do with a GWAS worth of variants like this? How is this helpful to us? Well, the first thing that everyone does is they're like, wow, you have 127 genes. That's so exciting. We're going to study every single one of those genes in an animal model. Well, one thing you want to keep in mind is that a GWAS hit is not necessarily a gene. And so each one of these hits actually represents a genomic region. This is zoomed in on that one I outlined there for TMC01. And there are many genes in the genomic region that is tagged by that SNP that reaches genome-wide significance. You really have to do a lot of fine mapping to know what the gene is in this region that actually is the gene that's contributing to disease. And I'm not going to say another word about that, because I know that Yutao is going to talk about that, and later on this week, Ayelet's going to talk about that, so you'll learn more about that. What I am going to talk about is genetic testing. So another thing you can do with this big, vast number of genetic variants is you can use them for genetic testing. And so when we test for early onset disease, it makes sense to test for a gene, find a mutation, and look at it in a family, and that's the disease. But for adult onset disease, like a GWAS worth the disease, each individual variant contributes just a small amount to genetic risk. So it doesn't make sense to test for each one singly. So instead, we test for a panel. And in recent years, this panel kind of approach, there have been algorithms developed to look at a whole genome worth of variants that contribute to disease risk through something called a polygenic risk score. And so here, there's just 4 simple steps. You take a GWAS, well-powered, you identify the model for calculating the weights for all the variants that go into the model. You then calculate essentially sum the risk variance in a person, and then you look at how that person is distributed against their population for how many variants they have. So some people are in, the very top of that distribution. They have way more variants than everyone else. They're like in the 90th percentile and have the highest risk. And other people, of course, have just a few variants, and they're in the lowest risk. So we've learned a lot about polygenic risk scores now for glaucoma based on these large genome-wide association studies. And we find that people who have glaucoma and high polygenic risk scores have more severe disease. They have earlier age of diagnosis by 5 to 10 years. They have higher intraocular pressure. They have thinner retinal nerve fiber layer thickness. They have an increased likelihood of progression and a greater need for surgical intervention. I think one of the things that's really exciting about the field right now in terms of polygenic risk is understanding, more about what other factors can contribute to modifying the risk. This is just one area of research that I think is a high point. And that is that when we look at, most of the time, people in the highest decile will have disease, but what about those people in the highest decile that don't have disease, or people in the lowest decile that do? And what we have found actually is that there are some metabolites and other kinds of environmental factors. Here's a recent paper that just came out that can actually modify that kind of genetic risk. So this is an important area for the future. I want to touch just briefly on genetic testing for early onset glaucoma. This is also a really important thing to be able to do, especially for these families, to identify people at risk in the families, inform genetic counseling, and establish a molecular diagnosis. One of the problems that we have right now, and another kind of current challenge, is that genetic testing only identifies a diagnosis in these people about 20% of the time, probably because we have such a small number of genes to work with. So there's a lot of effort right now in the field to working on ways to improve the genes new gene discovery to be able to identify additional genes that contribute. And I'm running out of time, but just briefly, I just wanted to say that There are some genes that contribute to both early onset and adult onset disease, but the mutations are different. So in this pathway, which is really important, the Tie2 angiopoietin-1 pathway, almost every protein in this pathway contributes to disease, but they contribute to a full phenotypic spectrum of disease depending on the variants in the gene. And so it can be adult onset disease, early onset disease, et cetera. So just to summarize, human glaucoma is highly heritable. There are 12 genes known for early onset disease. Most of these are involved in development of extracellular matrix. Genetic testing is useful but is limited. GWAS for adult onset glaucoma with complex inheritance has identified many, many disease loci. Polygenic risk scores based on those GWAS results can identify glaucoma patients with more severe disease, allowing for risk stratification, modification of surveillance and treatment. There are factors that can modify PRS risk, and that some of these genes can contribute across the phenotypic spectrum. I have a whole bunch of unanswered future questions, but we can talk about those later. And then I also have some references in the talk.
Janey, thank you for taking a very complicated and highly advanced topic and boiling it down into 12 minutes. I appreciate that. So I'd like to introduce Yutao Liu. Yutao is at the Medical College of Georgia and Augusta University. And you're going to talk about genetics and future therapies for GWAS data.
Thank you to everyone. So really, my great honor to be here to share something with you over the last 20 years we have done so far for the glaucoma part. So really, I have no disclosure right now. And so really thanks to Janey already showing you the Manhattan plot. And so look at all these, different. This is a different plot, but for IOP, published in 2018 from Anthony's Stewart study. So there's over 120 some loci for eye pressure. So I'll give you some examples, really how to do this part. There's really a combination of different technology together. It takes the whole community to work together. So in this way, a few years ago in 2018, so our group started looking at all these different loci, and then based on the data we have over the years from working with Mike Hauser, Rand Arlingham, and also Janey Wiggs and Lupus Coley, over the years we have human tissue, human cell. We did a microarray expression. We did different sequencing together. So we designed this pipeline to look at the genes expressed in glaucoma-related tissues, and then also combined with bioinformatics tools, including gene ontology and pathway analysis network, upstream, downstream regulators, and so really try to put all the things, also gene correlation, analysis in different tissues. So at the end, to our surprise, we identified actually estrogen receptor signal is one of the target genes. So here, all the gray, shaded genes are the IOP-associated genes. So we take these genes from the GWAS, and then we identify and fill the gap how these networks are closely related. We just try to identify the low-hanging fruit in these networks. And then we look at a little bit further and so say, okay, yes, many of these IOP genes, CAV1 and ESR1 are targeted. And CAV1 from the genetic association back in 2013 from Janey and Lupus Coley study is really affected mostly female with signal situation. So this is kind of fit perfectly. That's kind of a indirect validation of our finding. Yes, and also many of these genes also targeted by dexamethasone. This is connecting the biology together. So then after this, we look at the estrogen receptor knockout mice. Unfortunately, there are 3 receptors, ESR1, ESR2, and G-protein coupled estrogen receptor 1. So I'm going to talk about that data on Saturday if you stay long enough. So we do see IOP elevation in those mice. So really some are sex-based relevant, some not, but this really opens our new window to look at this. And also lots of literature data on this, and Hannah Youngblood and we wrote a review article together on this estrogen and glaucoma, and also Andrew Fiola here also studied this. Everyone in the community already know estrogen and menopause is really play a big role in IOP regulation and glaucoma risk. So really, that's just one of the things we want to share. And the other way is about the targeted genomic DNA sequencing. So this really to see all the genes there, which one is really causal, and then really to design some gene functional specific assays related to the glaucoma. So this is why, Yang O at the beginning set up the tone for glaucoma physiology, pathology. So it really depends on everything. So, and all the animal models we have at the beginning. So you, thanks to Janey, already showed this Nature Genetic paper published in 2018-17, showing the LOXO1 association there is really high, accounting for over 95%, so in the Caucasian population, over 80% of the individuals are carrying, homozygous variants there. So what's the really causal variant? So led by Ting Ang and CC Crow at Singapore, so really look at this gene. So the genomic region of LOXO1 is about 40 kilobases. And so they sequence this in 10,000 cases and controls. So look at all these variants. They identified one of the common variants, the rare variants, but relatively common in this population. So it's a coding variant in LOXO1. And then also look at all these different haplotypes from multiple variants. You combine this. At the beginning, you can see this table is a TGA or GGA. So that's T is really, so different risk variants go together. That could regulate the glaucoma risk. And so they really designed the assay to look at the cellular adhesion strength in the cell culture dish, and with over 40 different variants and haplotypes together. So you talk about someone, postdocs work on this, clone everything into cell line, you do all the assays and quantify, and to really look at all the variants you found. Are they functional or are they not? And so these are lucky we found all these coding variants. So this is one of the things. How about the ones that really are not in the coding variants? So she has done a lot of in this area. So 95% of the coding, the variants identified through glaucoma GWAS are not in the coding area. They are intergenic or really intronic. And what else we can do? So there's something we can look at those variants and their alleles. Are they correlated with gene expression in different tissues? And then, also Janey and others have done really SRI knockdown in zebrafish, because you can do the embryo. So within 72 days, you can look at the embryo of the zebrafish and look at the ocular phenotype there. And also, people like Simon Zhang, when he has unlimited funding at the beginning, he can knock down every IOP-associated genes. That's before 2020, when we have only less than a dozen genes. And once Janey identified over 127, I don't think he can catch up. And also, one thing is, we know glaucoma is complicated, yes? And so even people with elevated IOP over 20 years, they don't really develop glaucoma, yes? And so really it's, a lot of things here. So one thing in the community is you all heard single cell, single nuclei RNA sequencing, yes? You probably heard single cell portal and also Charles Zuckerberg's, the Human Cell Atlas initiative. And so all these data are sitting there waiting for everyone. And so just to use one example here, so Dr. Joshua Sen's group from Harvard, they look at, they are funded by the Charles Zuckerberg initiative, and they look at, many human eyes and the anterior segment, posterior segment, and they are really the first one to establish this. And so, and also Dan Stammer and the other group also did the similar work. And so, basically, this is really showing all the anterior segment and posterior segment. So you have to do enough cells to represent all the cells possible. So you have to do really, over 1 million cells in there to see these things. And so you identify markers for every, potential cells. The good thing for the animal, model research community is to generate potential biomarkers for TM. And so right now we only have mycelium really, driven. So MTP-driven crease for TM. We probably need another one for sure. And so this is, I think there's a lot of data, and then ATAC-seq and DNA-seq data that look at the open chromatin area in the genome, especially in the TM. And so we don't have anything in the retina. AMD field has done a lot of work in this from the NEI. And so we really need to do a little bit more. So another part is glaucoma is not a disease only affecting the eye. And it's actually a window, look at maybe Alzheimer's or the other diseases. So there is a whole body mapping project called the Human Reference Atlas. And so here, just give you one example. So from the many things here, they look at all the organs. Fortunately, eye is one of the tissue here. And the next one I'm showing you is not, eye is not part of that. So this is really funded by Charles Zuckerberg Initiative, CZI, and the other is really the MRC from UK. So but you can look at the eye there. There's only 3 branches there. It's not funded widely. So if you have some billionaire friends, so convince them eye is important, because we lose vision is easily go to depression, and so a lot of patients willing to treat for lose a limb than the vision. And so the other part is really the genotype tissue expression project. So she is really a leader on this, and so they have collected over 1,000 postmortem adults with 54 different tissues. And so this is really, they did whole genome sequencing to have every common and rare variants there. And so really, then you have So at the bottom here, so you have all the different assays. So RNA sequencing, epigenetic mapping, look at the open chromatin region, histone modifications, acetylation, and so DNase assay. And you look at all those cells, they are tissues. There are no eye tissues at all. Even retina is not part of it. And so it's a kind of shameful, and so really we need to push the community, and this is why every year BrightFocus will organize this the advocacy day. So I really encourage everyone to consider to attend every year to let the Congress people know, representatives know, eye research is really important. And so with all this, and then also we talk about 3D culture, and then Jason talk about retinoids, and then organoids and organ on chip. I want to share with you one recent paper published from an investigator from Columbia University. And this is really one of the organ on chip. And so you use a PDMS, kind of like a gel-related material to produce these 2 chambers. On the temp chamber, you have the acellular channel, and then you have a cellular channel. There you coded, as the step 1, 2, 3, and they use actually acupuncture needle to code those cells, TM cells, and then Schlemm canal endothelial cells in collaboration with Dan Stammer. And they look at, so you put also aqueous humor in the acellular chamber, and then it will leak with the ocular perfusion through the perfusion process, and then it will transport it to the collecting channel there through the cellular double layer. So with that, that's really kind of very, primitive. And so in summary, it takes a lot of things and years to work on this. For example, after 7 years, we still work on estrogen pathway, but I think it really identifies something, now to hopefully to benefit patients. And also, we try to build, 3D, organ on chip to mimic, TM cells, Schlemm canal endothelial cells together, and to have environmental factors. We can even flow the aqueous humor samples from glaucoma patients versus controls. So this is really the other way. Take-home message. And so it's really, I want you to remember, so GWAS, you get many genes, 300 genes or more. They are not causal, because one of those is your blood type, ABO blood type. And then glaucoma is multifactorial. And so it's not really rely on one single gene model. So the best model to use basically combine all the things together. Some models may tell you something is potential, some is not, and so we always need to validate, the models we actually use, and using human patient tissue or samples. So there's a few unanswered questions. I think really which gene are really causal. This will take the whole village. And then really to look at gene-environment interactions. And so, yes. So really combine in vivo, in vitro models, doing that together. So really the genetic impact is not going to determine the pharmacological impact there. So yeah, these are the reference. Yeah, thank you very much.
You know, thank you, Yutao. We certainly have our work cut out for us over the next years. So our final speaker is Ahmara Ross from the University of Pennsylvania and Shea Eye Institute. And she's going to tell us about the challenges of gene therapy.
Thank you so much. I want to use my time, and it's not the clock's not rolling yet. I just want to, perfect. I was going to use my time to make two really critical comments. I know that the room has become noticeably more sparse, but I vividly remember sitting in those seats during the BrightFocus Fast Track in 2017 and hearing Dr. Wiggs' first GWAS discussion on the complexity, the genetic complexity of glaucoma. And so don't underscore the importance of what you're hearing today, and that each person is giving a presentation which is a taste of what is available for bright minds like you to evolve the field. And so anytime anyone goes to a smorgasbord, you take a taste and then you decide what you want to commit to fill your stomach with. The only way to commit to that is to ask questions. And the beauty of this particular conference is that you eat meals with the people that are speaking on these panels today. And so I encourage you not just to consume the tastes that we have for you today, as well as the tastes that are for the rest of the week, but to use that meal time to go outside of your comfort zone and talk to people that you might normally not talk to, and expand the field with your brightness. And so I'm here to talk about some challenges of using gene therapy in glaucoma. And do I push? I have some disclosures. The only one that I really want to stress is the importance of the NIH and NEI. I love the NIH and NEI. You should love it too, and you should do everything you can to advocate for its continuation. I don't have to go through. Dr. O gave you probably an absolutely fantastic background on glaucoma and how important it is to study it. It's a huge leading cause of blindness worldwide. It affects a ton of people, but clinically, it's a silent thief of sight. And so that's why you're hearing a lot of people give presentations about better ways to understand the mechanism of the disease, but in this particular panel, better ways to identify high-risk individuals before this disease silently steals their sight. You will also hear that the retinal ganglion cells and the retinal ganglion cell layer are the critical cells that demonstrate survival. My lab looks at retinal ganglion cells, but I also want you to encourage, as you're thinking about some of these genes that affect neurodegeneration, that there might be other cells like astrocytes and glial cells that are contributing to this particular disease phenotype. I always love the opportunity. I feel very fortunate to do basic science, but also see patients. This is one of my patients that I think I will continue to use in my talk to outline one of the major challenges of the field of therapy in any type, which is how slow and variable the disease of glaucoma is. This is a patient who I inherited as a fellow in 2016 who continues to have disc hemorrhages. She has disc hemorrhages at normal pressures. And so in panel B, you'll see in a human nerve what a disc hemorrhage looks like. This is a focal area of damage that you're able to then identify on structural and functional bases. Sometimes one preceding the other. And so in this particular example, the patient had a hemorrhage in 2016. It's documented. She finally showed OCT damages in 2020. and then later showed functional Humphrey visual field damages in 2024. And so when you're dealing with that timeline, boy, therapy outside of IOP is challenging. But the vision loss is slow and it's irreversible, so it's certainly worth studying. There's a strong association with IOP and genetics, as you've heard from the panel. There's the loss of retinal ganglion cells, and it turns out other cells are important, but most of the treatment is aimed at lowering IOP. That's the only modifiable risk factor we have. And that brings me to the other issue, which is that there's a huge issue with compliance clinically. And so this sort of enters into how my interest in gene therapy to treat the disease began. The treatments, when you focus only on IOP, it ignores neuroprotection, it ignores our understanding of the pathophysiology of disease, and it ignores the opportunity for disease reversal. As I mentioned before, blindness clinically is often caused by compliance and non-adherence to medications or doctor's appointments. And then the therapy is often limited by the time progression and variability of the disease. I will primarily focus on number one in some of my therapy, but I'm sure some of my presentation, but I'm sure there will be questions that will broach the subject of others. Gene therapy, it involves replacing, inactivating, repairing, or what my lab studies enhancing a pathway to treat a disease or condition. But I think there's some considerations for the pathophysiology of disease that a lot of work in the neurodegenerative space has taught us, which is that before you think of just simply replacing a gene to treat a disease, you have to put some thought into how that gene is contributing to the pathophysiology of neurodegeneration. You can have loss of function. This is when a mutated protein has its function completely abolished. So this is one of several studies in dementia, where they are looking at loss of presenilin activity that contributes to age-related neurodegeneration. And so in this particular example, you have on what is your left side, a wild-type animal, and on the right side, a presenilin conditional double knockout animal. And when that gene is doubly knocked out, you get atrophic changes and decreased thickness in the cortex of animals compared to the wild type. And so this is an example of where you have a loss of function that's contributing to the phenotype. You can have gain of function where a mutated protein acquires a new or a toxic function or becomes overactive. This is an example of another type of neurodegenerative disease, ALS. On the bottom panels, you've got H&E sections of non-transgenic and then the transgenic animal over time. And what you are seeing over time in the transgenic animal is that you're getting loss of dopaminergic neurons by about 6 months. That's contributing to some atrophy. Let me see, is there a way to? Okay, good. Right here. Right here. And so when you express the normal protein, you get acquisition and buildup of some of these plaques. You can see that in the middle. And then when it's expressed with the mutant and the wild type, you get more of those. And so this is an example of where you have the mutant that acquires a toxic effect, causing a neurodegenerative phenotype. And then finally, dominant negative. This is when mutated protein interferes with proper wild type protein function. This is an example of where simply providing the wild type may not be enough to overcome the phenotype. And I want to keep going on in time. So these are just two mechanisms that I think about when I think about gene therapy. I always like to present this data. This is data out of my lab that looks at SIR2 It's not replacing a wild type protein. It's not CRISPR correcting a mutation. It's what I like to call the Monty Python protein. beginning in 2017 and even before that, in collaboration with Ken Schindler, we were able to demonstrate that enhancing the sirtuin pathway was able to preserve retinal ganglion cells in multiple models of neurodegenerative disease. This is one example of us using it in human cells. And here we are showing that when we treat these cells with a toxin that causes decreased survival, if we overexpress the sirtuin-1, we're able to improve survival as well as improve function in those cells in a dish. I call it the Monty Python because people may remember the scene where the guy is like fencing and he's doing all of these gymnastics to try to fence this guy, and the guy just pulls out a gun and shoots him. And so I like to call that my loaded gun. But that's something we're doing in the lab, but other labs are certainly using these pathways as well. This is the calcium calmodulin protein kinase pathway. This is not research done out of my lab, where they are using NMDA as a neurodegenerative mouse model. They overexpress using AAV the proteins that are enhancing the calcium calmodulin pathway. And in this particular optic nerve crush model, they're able to show in control, they get loss of retinal ganglion cells, but in cells that, in mice that they have designed to overexpress proteins of the calcium calmodulin pathway, they're able to improve survival of those cells in crush, even up to 6 months post-disease. And finally, I mentioned dominant negative effects. This is an example of emphasizing that glaucoma is not just a disease of the back of the eye, it's a disease of the front of the eye. This is a mouse model of myocilin, where they have a mutation that causes a high pressure and cloudy cornea from the disease phenotype. They CRISPR corrected it. They were able to address the elevation of IOP that's seen in the mouse model, the deposition of myocilin in the trabecular meshwork, as well as they were able to reverse through lentiviral delivery and CRISPR gene editing of that particular mutation. Finally, something else that I personally am very excited about is work out of Stanford. Wang Hu, Dr. Hu, looked at gene therapy and controlling expression of therapeutic genes in healthy versus unhealthy neurons, these so-called disease-sensing promoters. So in addition to being cell-specific, it can be disease-specific. And so I'm very, very interested about the research that's going to come out of that lab in the coming months. Some other considerations that I haven't gone over, there's a mountain of work on gene delivery and vector techniques. There's naked nucleic acid delivery, lipid nanoparticles, insert your delivery du jour. That's an area that's ripe for research. There's a growing body of interest in your immune response to these viral vectors. Certainly, we can address the issue of compliance by delivering a therapy to the cell type of our choice, and perhaps in a year, in the presence or absence of disease. But if your immune system eliminates that particular therapy or the cells that are expressing your gene of interest, then we end up with the issue of compliance and needing to re-deliver. And so that's an area. And then regulatory hurdles. I have one more slide, but I had mentioned before the timeline of neuroprotection. That was almost 10 years where you look at a focal area that shows damage. And so if you want to look and see if your therapy is effective, there's a growing body of research going into addressing some of the endpoints of success for neuroprotection, addressing some of the endpoints of success for gene therapy and delivery in human disease trials. Despite these challenges, there are tons of human gene therapy trials in effect. There are even more than when I made this slide. And so I think the field is ripe for bright minds like yours. In summary, this is disease with tons of basic clinical and translational questions that are unanswered. I think gene therapy can offer a paradigm shift, not just at understanding the mechanism of human disease, but potential therapies, and there are tons of unanswered questions. Here are my take-home messages, but more importantly, Don't rely only on IOP models if we want to look at our efficacy of response. Don't ignore immune response. Don't assume a uniform disease progression. That's not how it happens in humans, and underestimating delivery challenges. My references are on this QR code. I've downloaded some PDFs just in case there are any issues with downloading, and I'll happy to take any questions.
Thank you, Ahmara, for a wonderful talk. So we're open to questions from the audience. We wore them down. Okay, we got two. One here and one over here.
Thanks so much for your talks. A fascinating topic. We talk about this at work a lot. I'm on the drug development side of things. And when we think about gene therapies, we often battle at work because we think about these diseases as multimodal. And I always wonder if we're looking for the holy, and I'm coming from geographic atrophy, so my glaucoma is a little less put together. But when we think about this, are we trying to find the holy grail in disease to understand the mechanisms, or are we trying to find something that we can use to treat, or both? And I just wonder about that because I come from schizophrenia as well, and we call it polypharmacology. You throw every drug at every receptor in those diseases, and it works. Whereas if we're looking for one target, how do we drug that? And the second part of that is thinking about age-related diseases, and on a different side of things, how do we get payers to pay for gene therapies for this population of people? So a number of questions there, very exciting topics, and we'll love to hear your thoughts.
Wants to start?
You go ahead, Ahmara.
That's a lot of questions and it packed into one question, but I think the eminent things that come to mind to me are, I don't know that we're looking for one holy grail. I think we learned from a lot of our colleagues in the inherited retinal disease world where you're trying to treat individual gene mutations that might be contributing to a phenotype. And I think scientifically, that makes a lot of sense. Scientifically, it's absolutely, it grows the field, it helps us understand the pathophysiology of disease, but boy, I think that particular field could benefit from perhaps a gene therapy that could be used for an umbrella, and I think that field is also moving into there. I think in geographic atrophy, some of the challenges for you was that the damage is pretty done. And so once you get to the point where you're treating geographic atrophy, the space that you're really looking for is probably cell transplantation, which is a little more, less translational and more basic at this point. I hope that answers some of your questions.
And I'll just add one point. It's going to depend on the gene and whether it's a dominant negative.
Yep.
For example, with myocilin, you can knock out myocilin in the mouse, no phenotype. There's humans running around that are myocilin null. They don't have a phenotype, at least that we can detect. So the strategy there is just knock out myocilin, and it doesn't matter which mutation is causing the misfolding, right? So it's going to depend on our understanding of the disease, which is what you had said. Yeah. So both understanding the pathogenesis and then coming up with a way of treating. Brian.
Thank you. My question, I guess, is to the panel, but was triggered really at the part of the talk that Janey Wiggs was giving about polygenic risk scores. And we think about the two sessions together, machine learning, deep learning, AI, et cetera, and it's a very ignorant question, but when we get enough cases that have had a polygenic risk score and then subsequent, let's say, rates of progression, you talked about the weighting, that you had a four-step model, and weighting, I think, was step 2, to create a model for what the final risk score is going to be. Is there room for machine learning to go back to reweight those to make the prediction stronger?
Yeah, actually, that's not what I thought you were going to ask. But so There are a number of algorithms for that step, and actually, that step is important, but and certainly there is benefit to focusing in on those weights, but it's not as critical for primary open-angle glaucoma, at least not so far, because most of the genetic variants have kind of similar effects. If you were, tackling something like exfoliation syndrome, where you've got to deal with this gigantic LOXL1 issue, then that's a little bit of a different thing. But let me answer the question that I thought you were going to ask. I think the integration of AI and perhaps imaging, or AI of clinical data with polygenic risk scores is going to be really useful going forward, because what we get with the polygenic risk score is very important. We get a batch of people that have high genetic risk and are likely to have more severe disease. But what we'd like to be able to do is know what is the best way to identify that early stage disease in those people. Remember, the ultimate goal here is that we identify people who have high risk before they have disease. And then we have a way of approaching them clinically so that we can identify people that will benefit from the earliest possible, new therapies as they come online. And so that's really where I do see an interesting opportunity to integrate AI with genetics, is to build this kind of model that is genetic, but also is based on important interpretation of clinical features that we can use AI for.
I also think, just broadening on that, the idea of using AI to understand rates of progression so that when we implement therapies, we can see if that rate is changed in a significant way. I think part of the limitation with therapy is that the disease is so variable, it's difficult to identify a noticeable change in a patient to see, oh, this is working. This is bromonidine, I'm just giving you an example, is working. And it's tough to see if that delta is the variability of the disease or if that's the actual therapy that's contributing to a plateau in progression.
So, John, yeah.
Largely for Jamie. So the polygenic risk scores, I love these ideas. This concept is fantastic. But one of the things that come to mind is when you find so many SNPs that have very small effects, the frequency in the population might be very high. Is that the case, what you're finding, that these frequencies of these SNPs are pretty frequent and they're just not associated with disease very often? Is the first question.
Yeah, okay, so individually they're high.
Yeah.
Individually you see them in 30% of the population. That's why testing individually is not helpful. Because 30% of this room would have a high risk factor for glaucoma. But the real beauty of the polygenic risk score is that you're looking at an aggregate effect, which is, you can make it as rare or common as you want, but if you look at people who are in the top percentile of that distribution in most populations, they have an excess of genetic risk factors that leads to a high genetic risk of disease. And that's a meaningful test. So it's meaningful to be able to separate then those people from, the rest of the distribution who have normal to low polygenic risk scores. There's a benefit for testing those people as well. I think Yvonne this morning noted that, to be able to identify people who don't have high risk. That's a very useful thing to do also. But yeah, the advantage of the polygenic risk score is that, you get a group of people who have high genetic risk for disease. They're highly likely to develop disease.
So one of the surprising findings from the GWAS studies for AMD was the protective SNPs as well. Were those pretty common in the glaucoma GWAS studies?
Yeah, so there are well, so every SNP can be risk or protective. It just depends which allele is more common in the population or more common in cases versus controls. But interestingly, we do have a major locus for POAG and also associated with normal tension glaucoma, where the common alleles, the ones that would be 30% in the population, are protective. And so, you know, and there are populations with increased risk of disease where the allele frequency for those protective variants is lower. And so there is that kind of opportunity. There's also some rare variants that are protective. Angiopoietin-like 7 missense alleles are also protective. And Anas Abubakr is right over there, has studied these alleles, and that's a really also an interesting story.
So, Mike?
Any insights on how we can do better on the missing heritability of childhood glaucoma?
Okay, you want to read?
The question was how we can do better on the missing heritability of childhood glaucoma. That was on that slide that I had to skip, you know, because of the warp speed of the timer. So yeah, it's a conundrum. It's because, you know, why do we not have more genes for congenital glaucoma? We really should have more, you know. We've been doing this now for almost 3 decades, and we have 12, if you count every single last one. So I think there are several things. One important thing is that a lot of the previous studies have been in European populations. And, you know, so if we look in some other more ethnically diverse populations, we might find other genes. And that actually was the strategy we used for EFEMP1, and indeed we found that. Another thing that's interesting is that most of the screening has been done looking at coding sequences, you know, exome sequencing, Sanger sequencing. And I think that as we transition more to whole genome sequencing, it gives us an opportunity to look at things like that are adjacent to coding, like splice site variation, which, you know, sometimes you pick up and sometimes you don't. But even more importantly, copy number variation. And, you know, we've just been involved in an interesting study where, you know, a duplication in FOXY1, which is one of the well-known genes, which has gone pretty much undetected, is a relatively common cause now of juvenile open-angle glaucoma. So I think we'll do better, you know, but I don't think it's going to completely solve the problem. And I do think we have to widen our thinking a little bit to things like complex interactions. You know, are some of the juvenile people actually just people at the very extreme of a polygenic risk score? So we need to think about it more.
Yeah, and I think we also need to start thinking about epigenetics and how epigenetics can be playing a role in the pathology.
This is mostly for Dr. Ross. I guess mostly my question is, what do you see as generally the biggest bottleneck in directing IOP, non-IOP directed treatments from the clinical or from the lab to into the clinical pipeline?
Yeah, I'm interested in what people in the audience think that also work in this space, but I know for me, a lot of our models are high IOP, and so we find things that can prevent degeneration of retinal ganglion cells or reduce bad astrocytes from coming in and causing degeneration of disease. But there's not great models, barring anything that was discussed at this past panel, of normal tension glaucoma. Because I don't know if in the clinic someone's going to say, well, we just won't treat this pressure. Let's treat with this therapy and see how it goes. That's number one, I think. And also, I am interested to see how the organoid and human cell space evolves in this field in terms of trying a bunch of therapies and having clear endpoints and outcomes of success to go into larger animals and even human trials, because I feel like it stops at the mouse model, and then we never really get any of these really great therapies past that point.